Welcome. Thank you so much for joining us today for, I think it's now our fourth talk in our Borderlands Technologies speaker series. We really appreciate everyone's participation and the discussions that we've been generating in this space. So today we have a representative from the Fundación de Antropología Forense de Guatemala, or the Guatemalan Forensic Anthropology Foundation. It was supposed to be Michelle Stevenson, who is their director of their genetics lab, but unfortunately there was a last minute emergency in one of their projects and she's on a plane to Colombia, which I think really speaks to the important profile that FAFG has in Latin America for forensics. And we are so lucky to have Wendy Argueta instead. Wendy and I actually have known each other for a long time. She trained me in some forensic genetics methods some 10 years ago, along with several other forensic people from across Latin America. And so I was just so thrilled that she was going to step in and speak with us today. So Wendy is also a DNA analyst level 3 for the Forensic Anthropology Foundation of Guatemala. She's been working in forensics for 11 years now. She has her undergrad degree in biology and biochemistry and her master's degree in forensic genetics from University of North Texas in the United States. And she works and lives in Guatemala, and is going to talk to us today about multidisciplinary approaches to the search for and identification of disappeared people. We welcome questions. Please use the question tab or the chat to do those, and it's fine if your questions are in English and Spanish. Wendy is going to give her talk in English, but she's happy to converse in Spanish during the question and answer time period. So I'm going to turn it over to Wendy. Welcome.
Hi, thank you very much. So I'll share my screen and please let me know if everything looks fine.
Looks great, Wendy.
Okay, thank you. So good afternoon, everyone. I want to thank you for the invitation to participate in this event. It's a pleasure to be able to talk to you about the importance of working towards the search and identification of missing persons. So I'll start talking to you a little bit about our history, Guatemalan history. And an armed conflict took place in my country from 1960 to 1996. It was a small leftist insurgency and a devastating response by the state, leaving an estimate of 200,000 victims, civilian victims, from which 160,000 of those were killed in their communities, including small children, men, women, and even the elderly. And we also have 40,000 missing persons, the ones we're still looking for today. According to the Truth Commission, 83% of the victims belong to Mayan population, and the other 17% to non-Mayan victims. Now it has been determined that the majority, the 93% of the crimes, can be attributed to state forces, including the police, the military. The families, they want information. They want to know what happened. They want the bodies of their missings, but they also want to let everyone know that their loved ones did nothing wrong. So in 1995, the FAFG was founded with the mission of searching, locating, recovering, and identifying the bodies of the victims from the armed conflict. The reason to start fulfilling this search begins with finding their loved ones alive. But if this is not the case, then recover the bodies. It also allows the family to secure rights of truth, reparation, justice, so that these events are not repeated ever again. FAFG fulfills this mission applying 4 technical disciplines that are part of a multidisciplinary system or approach that helps to assist families in the search for truth, justice, and recovery of the remains of their loved ones. At the end, the use of all these 4 scientific techniques and procedures allow to carry out the analysis of physical evidence and report objective and accurate results. Even though our mission is to identify missing persons from the internal armed conflict, this multidisciplinary approach can be and has been applied in any other context or events, such as wars, natural disasters, drug trafficking, and of course, it can be used for migration investigations. So the first department is the victims investigation and documentation. They are in charge of locating and reaching out the families. The families are the ones who can give us as much information as they can and remember about the missing person. During this interview, what we call antemortem interview, the investigators ask the relative everything they can remember about their missing one, such as age, height, physical features, especially those that can or that are individual characteristic for that person. We can mention a fracture, a surgical intervention, a dental job or modification, anything actually that can be unique for that person. So if the body is recovered, it is useful during the identification or comparison process. In the case of migrants, information about tattoos, visible marks in the skin, moles might also be useful because probably those features will still be visible on recovered bodies and can be used in the comparison. In addition, this interview also claims to collect information about the event. What happened? When did it happen? Where did it happen? How many victims were involved in the event? And in the case of migrants, we can think of information such as where were they going, which was their final destination, when did they left, really anything that might help to locate the missing one. They also collect the DNA sample. For us, it is either a buccal swab or a blood sample collected from the family donor. This step also includes filling out the informed consent that informs the donor that all information shared with us is confidential. This type of information also includes their genetic information. It also explains that the DNA sample can only be used to carry out DNA comparisons with the purpose of identifying their missing one, and they are asked to sign it if they agree and if they understand the purpose of this sample collection. The DNA sample is then packed in these paper envelopes. They are correctly identified and sealed to be delivered to the forensic genetics laboratory. Since DNA comparisons is one of the identification disciplines included in our approach, the biological relationship between the family donor and the missing one needs to be correctly documented. And this is done by building a genogram, which will later be used to define the family structure, the number of available donors, and the individual relationship to the missing one. We might face scenarios where the family members are living in different departments here in Guatemala, but it can be states or even countries. So it is likely that contacting them and collecting the samples all together may not be done at the same time. So this diagram or this genogram also helps to register if other family members are available as possible donors that can be reached and asked to donate the sample in the future. Some family groups, such as the one that we're looking here, might have more than one missing relatives. Those are represented by the sky blue squares in the genogram. And the family reference donor will play different roles for each victim. And this has to be correctly reflected in the genogram. As more family references are collected, more genetic information is available for the comparison. And this is crucial, especially for those cases where only distant family donors are available. One of the most successful strategies that FAFG has used in the past to reach families has been the use of advertising in billboards, bus stops, and local radio stations, letting them know that by giving their DNA sample, their loved ones can be identified. Anytime this type of advertising was released, the number of people approaching FAFG increases. And even though this is by far a scientific technique, the possibility of publicizing the work to let them know about the existence of this effort is crucial, because without information about the missing person, information about the event, and the DNA sample, the rest of the steps could not or cannot be done. One important aspect to consider is the language and the media available or common used by the people we're trying to locate, because not everyone will talk the same language, and they will have access to different type of media. So that's something to consider. Then we have the forensic archaeology department. The forensic archaeologists are the ones in charge of performing exploratory steps in order to locate mass graves. And once a grave is located, they move on to excavate it and exhume the remains. Usually when a crime occurs, there is a crime scene that will be analyzed, and all possible evidence will be recovered to find out what happened. In our context, the common graves are the crime scenes containing that evidence, which has to be carefully recovered and documented. Everything that's found inside a mass grave is registered in an archaeological record and photographs. You can imagine that this is a very thorough work that has to be done in a way that the evidence doesn't get destroyed during the exhumation. They also document another characteristics of the grave, such as the depth, and they can conclude if that is the first burial site or if it is a second burial. They can also determine if it was an individual burial, just with one remain inside the grave, or if it has multiple human remains inside. This, for example, is a grave where 64 human remains were exhumed. You can see that the remains are piled up. So it can be considered that those bodies were thrown into the grave instead of being placed in the grave. The archaeologists have vast experience because they have conducted investigations in different types of soils, in the field and in cemeteries. Most of the remains recovered in our context, in FAFG's context, are in a state of skeletonization. But they have worked on other cases, more recent cases, where bodies are found in earlier states of decomposition. The recovery of the remains needs to be done with respect. And here you can see that most of the times, anytime possible, the families and people of the communities are present during that step of the process. We know and we realize that this cannot be done in every single setting, but we believe that it is important to include them in this part of the process too. Then all the remains exhumed are transferred into the forensic anthropology laboratory, our third department. Here, the experts are in charge of analyzing every single bone recovered. And this analysis includes determining the biological profile of the remains. This includes trying to determine the sex, the age, and the height. All information recovered from the bones, it's documented in this anthropological file, and also by X-rays analysis and photographs. During the anthropological analysis, three types of trauma are also determined. The antemortem trauma, all those that took place during the lifetime of the individual that might have a mark or a scar in the bone that can be unique and help in the identification process. They also analyze post-mortem trauma, all those traumas the body might suffer after death, and circummortem trauma, those occurring close or during the death. Sometimes this type of trauma can also explain or give information about the cause of death. Here you can see a picture showing a wound left by the impact of a firearm projectile. And in this picture, you can see several wounds, most likely caused by a machete. Items such as coins, documents, clothes, or any other item found associated to the individual are cleaned and also documented. So in summary, the anthropological analysis is very important, and it can reveal a lot of information from the body or the skeleton recovered, along with other items that can be found related to the victim. And finally, the last step is to collect a bone sample or a tooth sample from the skeleton remains, and these samples are delivered to the forensic genetics laboratory to be analyzed. So fourth department is the forensic genetics laboratory. And in here, we work with two types of samples. The skeletal, the one that is shown in this picture, which is referred as the unknown sample because we don't know who that DNA belongs to. And we also have the reference or known samples, those collected from the relatives. These are known as reference samples because we know who the donor of the DNA is. These two types of samples are required in any type of investigations that includes DNA as one of the identification disciplines. If any of them are missing, then the comparison and therefore the identification cannot be achieved through DNA. Known samples can be out of two types. Samples collected from someone biologically related to the victim or to the missing one that will be used in a kinship comparison. And the second type are direct samples, meaning samples containing DNA from the person that we're looking for. Let's say a toothbrush. Direct samples from the victims are very useful in the comparison. And they even make the comparison a lot more easier because basically the genetic information in the unknown sample and in the known sample is the same. But they are not always available, especially in cases like the ones FAFG works on, where events happened 30 or 40 years ago. My guess is that in migrant cases, both types of samples will and can be available to work with. And this brings another consideration. And this is that forensic genetics laboratories need to be able to process the variety of samples that might be collected during an investigation. FAFG laboratory has validated procedures to process buccal swabs, blood samples, and skeletal samples. Skeleton remains are one of the most challenging samples, so very sensitive methods are required to be able to recover and report DNA profiles from this type of samples. But we also have procedures to process other type of samples commonly collected from criminal cases, such as sexual assault kits, hair, nails, fabrics, or other items with blood or semen stains, just in case if we need to process those type of samples. The FAFG Forensic Genetics Laboratory works under a quality management system accredited under the ISO 17025 standard, ensuring that all methods and procedures used comply with the intended purpose, that all results reported are accurate, that all equipment and material works properly, that the facilities meet the requirements, that all registers are up to date, and also ensures that the personnel is competent to perform the work and report the results. Once a sample is collected, either a known sample from the reference family donors or an unknown sample from the skeleton remains, the first step is to obtain the DNA that is inside the cells. And this is known as a DNA extraction. Usually the DNA extraction step is followed by a purification step where everything else but the DNA is eliminated from that sample. Next, specific regions of the DNA called STR markers are tagged, and then they are multiplied until millions of copies are generated. Right now, we're using a kit that amplifies 24 regions of the DNA altogether at the same time. So these tagged regions are then injected into a genetic analyzer where they are separated based on their size and tag color. At the end, we end up with this type of digital information or representation of all the DNA fragments obtained from each sample. Basically, each one of these signals that you can see here represent a section of the DNA, giving information that it is unique for each individual. Here you can see that we have information for 24 different regions of the DNA. And for a sample collected from a single individual, we are expecting to see 1 or 2 signals at each region, 1 or 2 peaks. here. Sorry. So at the end, we are reporting basically a pattern of number that is unique for each individual, with the exception of identical twins. That is the only case where two individuals will share the same genetic information. And you can see in this table that we have all those 24 regions. That's the name in the dark blue line. And under that, we have the alleles or numbers. This is just the number of repeats. And you can see that we have two numbers per region of DNA. The only marker that will be reported using letters is marker amelogenin. This is a sex marker. So if we see an XX result, this means that this sample belonged to a female donor. And if we see an XY result, this means that the sample belonged to a male donor. And again, we will have two different numbers at each region. And one of these numbers is inherited from our mom and the other one from our dad. So basically, we share 50% of our genetic information with our mother, and the other 50% of our genetic information is shared with our father. And this is how comparisons through kinship works. We will share a percentage of our genetic information with people biologically related to us. This percentage will vary depending on how close or distant that relationship is. Now you can see the importance of understanding family genealogy and the relationship between the family references to the missing loved ones. And no matter the context, it is crucial to correctly record the structure of the family group and what their relationship is to the victim. In the recent year, the implementation of new technology called mass parallel sequencing has been taking place into forensic genetics laboratory. We got the equipment last year, and we've been working on validation process and implementation of this technology into our workflow. This instrument allows the analysis of 61 STR regions of the DNA at the same time. But its advantage doesn't rely only in the increased amount of regions of the DNA that we can analyze at the same time. Since it is a sequencing instrument, besides giving the number of repetitions, which you can see on the left side of the slide, it also provides the sequence of that DNA fragment. So differences in a single nucleotide or single base of the DNA, which are known as SNPs, can be detected. In fact, this kit includes 94 identity SNPs that can be used for identification purposes, in addition to 22 phenotypic SNPs that were designed to infer the phenotype of the donor of that sample, such as hair and eye color, plus information for 56 ancestry SNPs that can help us to determine to which of these population, the populations that you can see on the right side of the slide, is more likely that the donor of the sample might belong to. So in terms of migration, this information may have a higher value, providing physical and ancestry information for the unknown sample. Once the genetic information is obtained, the DNA profiles need to be compared. All family members from a family group are compared against DNA profiles reported from the human remains recovered. Now, we may have a case where one family group is looking or searching for one missing relative. But what happens when you have multiple victims and therefore multiple family groups searching for their missing ones? Then massive comparisons must be done. Massive comparisons allows to search for a DNA match between 100 or 1,000 DNA profiles from unknown samples and 100 or 1,000 of DNA profiles from known samples. And usually this has to be done using a software. We've worked with two different softwares. The first one is DNA View that we use to carry out a comparison of closed context cases, such as those where one family group is looking for one victim, or small cases And the second one is mass fatality identification system, or emphasis. And this is the one that we use to perform massive comparisons. In fact, this software was designed to carry out the comparison to identify the victims of 9/11. And here we also built the National Genetic Bank of DNA profiles from family and victims of enforced disappearance. And in our genetic bank, we have over 16,000 profiles for family groups or family members, and close to 5,000 DNA profiles from skeletal remains. And genetic databases are crucial, and basically you need to have one if you want to do human identification in a massive approach. But a topic such as confidentiality, administration, maintenance, national or local laws must be taken into consideration to create or build a genetic database. So at the end of all this work, we end up with a lot of antemortem information gathered from the victim's investigation and the genetic profiles from reference samples. And we also end up with post-mortem information gathered from the archaeological and anthropological investigation and the profiles from the remains. If these two sets of information match, then an identification can be reported. The analysis of all this information for a case is performed by an independent department, a fifth one. We can consider it as a fifth department inside this multidisciplinary approach. After an identification has been confirmed and approved by the identification committee, the family members are notified by FAFG staff members. And the return of their loved ones is made in a dignified way, following their wishes on how, when the burial will take place, and everything is done accordingly to their traditions. So basically, FAFG has gained some experience in the past 24 years. Thanks to that, we have been able to work on different projects around the world, putting in practice this multidisciplinary system. It is worth to mention that the events leading to the existence of missing persons can vary, but the steps such as the search, the analysis, the comparisons are constant in any investigation. It is a fact that every situation is different and might bring its own challenges, but the scheme and the foundations can be applied and adapted to a specific need. In Guatemala, we work, again, this is what I've been talking about, the identification related to the victims of the internal armed conflict. But we also work with the National Adoption Council, the CNA, to ensure that adoptions in the country are carried out under legal framework. Many cases of illegal adoptions happen in Guatemala in the past, including those that happened during the conflict. This type of cases include children who were adopted by foreigners and are now living in Europe and the United States. But now they're trying to locate their families here in Guatemala. In order to respond to this issue, their samples are being collected and compared into against profiles in our data bank. And these organizations that are carrying out these projects are NextGen and Raíces Perdidas, or Lost Roots. And we also work with commissions to search for missing persons in Colombia, El Salvador, and Mexico. In Mexico, we are working on different states such as Sinaloa, Nayarit, Jalisco, Guerrero, and Coahuila. And recently, we were asked to participate as technical reviewers of the identification of 16 Guatemalan migrants killed in Tamaulipas in January of this year. And the most recent collaboration is with DNA Bridge. This is a consortium of scientists, human rights advocates, and scholars convening to advocate for the development of an international protocol that sets out guidelines and best practices for a DNA-led approach to identify and locating the families of children separated by armed conflicts, disasters, and inhuman immigration policies in a secure, ethical, and humane manner. So this effort is mainly focused in the reunification of families separated by Trump's zero tolerance policy. Specifically for those children in detention centers in the US. The United States has seen a surge of more than 70,000 migrants family and 45 unaccompanied minors crossing the border just between October of 2020 and March 2021. This is a screenshot of a paper that was published in Journal Science, advocating so that the massive DNA comparisons can be used for the reunification of those children and their family members that might be even in other countries such as Guatemala, Salvador, Honduras. DNA breaches has some objectives. Here I have some of them. And the first one is to construct a DNA-led approach for reunification of families outside of government control with global applicability, establishing a scientific strategy based on the studies of the DNA, emphasize the need to reunite families separated as a result of the zero tolerance policy, highlight the need to safety for migrant families and children and solutions for separated families, and develop a DNA-led approach to reunify separated families since 2017, where reunification is safe and responsible, and with considerations of the best interest of the child first. So to achieve this, this consortium has a advisory group to apply all these DNA approach and technology. But also this group has people who can carry out actions other type of actions that will be required in this process. For example, third parties are considered to coordinate several steps in the process, including gathering non-DNA information, of course, collecting those DNA samples, but also providing legal counsel and psychosocial support. And this effort for sure will require the collaboration of several organizations. To carry the DNA tests or kinship comparisons, accredited forensic laboratories will be involved. And of course, families are a critical part in this process. Their trust needs to be ensured, and they need to be treated with respect and applying principles of cultural and linguistic competency and fluency. And they will also will decide if they want to provide a DNA sample or whether or not to reconnect with their migrant, with their child. And of course, there are many challenges related to the management of the DNA data among multiple nations and jurisdictions. So this has to be done responsible, following international guidelines and best practices, where input from experts, including families, advocates, scientists, and government agencies is crucial. If you want to know a little bit more about this project, this is the website that you can access. And in terms of migrants, FAFG has the capabilities to fulfill this work, including 30 years of experience working with families and specifically creating that trust with them. We also have the local and regional context. We know the language, including Mayan languages. We have access to the communities, and we have the personnel and logistics to connect DNA samples locally and internationally. In terms of the laboratory, well, our laboratory is ISO accredited. We do local sample processing, and probably it is done at a lower cost compared to other international laboratories. We also have the experience with management of large number of genetic and antemortem data. We already have the software to carry out massive comparisons, and we also have the experience to deal and to perform these massive comparisons. And there is also Friends of FAFG, which was established in 2011 to support the work of FAFG to raise awareness in the United States of FAFG's work in Guatemala. And it also connects with the Guatemala living in the United States and allows us to receive funds in the United States to carry out our projects. Migration identifications involve several facts, realities that we know that exist. These include, for example, that remains are usually discovered by people like people that was hiking or hunting or rescue groups or the border patrol. And once the remains are found, authorities get notified, and then these bodies are recovered or the responsibility of local law enforcement officials, which will transport the remains into the medical examiner's office of different states or countries. Then we also have that the bodies of the deceased migrants are often not found. And if they are found, identification in many cases is difficult since irregular migrants by necessity often travel without documents and their families may not report them as missing. We also need to consider that because of the harsh and unique desert environment and the remote areas where people cross, the use of non-genetic methods such as fingerprints or photo comparisons may not always be possible. Also, migrants who are afraid of criminal proceedings or deportation may intentionally seek to avoid registration. And this fear is also observed in their families members that usually do not trust in the government. We also know that the approach of national authorities varies in time, and also that there is an increased number of databases about migration and missing migrants, but they are isolated and accessible only by a small number of individuals or agencies. And of course, this type of investigations will require some efforts, including cooperative efforts to account for migrants who have gone missing or have died, especially of the governments of the countries. But we also may know that, and it's a reality, that they most of the times don't want to be responsible or even pay for those tests. Also understanding the extent of the problem, starting by estimating the number of missing migrants, because this is information that is unknown most of the times. One reason is that family members usually do not report their missings or their migrants because they are scared. And also to create the capacities to provide the resources included but not limited to forensic and data system capacities and procedures to locate missing migrants and or identify their mortal remains. So this is it. This was my presentation. And again, thank you very much for having me here today, sharing the multidisciplinary approach for human identification developed and used by FAFG.
Thank you very much for the amazing presentation. So we will be collecting questions. You can write them down in the Q&A or in the chat, either in English or in Espanol too. Yeah, but in the meantime, I was wondering about best practices or international quality standards regarding dealing with the relatives and the, the psychological aspects, respect, trust. So yeah, I wonder if you can maybe elaborate about what these practices will be in order to address and achieve a good forensic science at the end.
Okay, thank you. So basically, this is done by a different separated department. I work at the forensic genetics, right? So I've never done it personally, but I know that you have to relate. First thing, you have to relate to the family members, and you have to find ways to explain it with respect, because most of the times they are looking for the relatives alive, right? And then, so how do you mention that maybe they won't be found alive, but we will start looking for bodies. And that's something that you have to explain very carefully and with respect, just to not close for, let's say, close the doors, and that will be the end of the investigation. So that first. Then we have, we work with a lot of visual material, pictures, how a DNA profile works, how a dental record works. We have different colors, like a palette of colors, because, for example, we ask the color of the clothes that the victim was wearing the last time they saw him or her. So we have colors there so they can point out the color. And in terms of, like, for example, the interview material, we've seen that different agencies have different interviews, different questions. Usually they are long interviews. And so we've been trying to adapt or select the questions that will help in our context. And I think that that's something that needs to be done. Because, for example, for us, we don't need to record or document if the missing person had a tattoo, because we won't find any tattoos in our context, right? So that's a question that we don't need in our interview. But it might be useful, for example, in migrant cases, because the body will still be, you can see that marks. So that's something that I've seen, that the interviews, they are usually long, but they should be adapted to each specific scenario or context that you're dealing with.
Okay, great, thank you.
Oh, and one more thing, sorry, is the trying not to revictimizar, I don't know how to say it in English, the relative, right? For example, we don't want to go the first time and just tell them about the FAFG work and ask 2 questions about the missing person, and then come back and ask another 10, and then come back again and ask another 20, right? So, because that, it means that they are talking and repeating the same event 3, 4, 5 times. So we try to do it in a way that they are not revictimizados constantly.
Great, yeah, I understand it. So yeah, we have a question from Vivette. Thanks, Wendy, your work is fantastic. I have a question regarding the plausibility of massive comparisons. In a context like Mexico, where we have 90K disappeared and 40K unidentified remains, and the rate of identification is at best 37 bodies per month, massive comparisons seem like the only way to make an impact. But what kind of databases, in terms of size, number of OCHI, etc., will be actually required in order to make progress in these identifications?
Well, there is no minimum or maximum number. The answer will be as many as possible. So as many as possible family groups can be located and a DNA sample collected from them, they should go into the database. And every DNA profile recovered from a body should go also into the database. And of course, massive comparisons are perfect for this type of cases, when you have this amount of missing persons, right? But we're talking about a country that is huge. So a lot of communication and a lot of understanding between states and databases, because I don't know, maybe a database will be placed in Coahuila and another one in Yucatan, but family groups of missing persons found in Coahuila can now be living in Yucatan. So there has to be some connection and comparisons between those databases, or just have one national database that will have all these DNA profiles and then do the match. And we have to face a truth. We won't be able to recover DNA profiles from all unknown samples. Mexico has a really complicated scenarios, because in some cases, people have found a way to destroy all evidence, meaning tissues, bones, teeth, so there is nothing left. Or the DNA is so damaged that we are not able to recover DNA from them. That can be a possibility. So if you don't have a DNA profile from that body, it means that it will never get into the database, and for instance, it will never be compared, even though if you have the family group already there. So we will face that problems, right? We will have the family, but we won't have the DNA profiles from the remains. And if the families are not reached and a DNA sample is not collected, we might end up with having a DNA profile from a human remain inside the database, and it will never be identified because we don't have the known samples, right?
Sure. Thank you. So any other question or commentary?
Wendy, I have a question. Thank you so much for a wonderful talk. One of the things that, so I wanted to ask you a little bit about, the talk is focused on multidisciplinarity and how there's lots of different departments at FAFG, which I think is one of the hallmarks, one of the really unique aspects of your work. And from the very beginning, my understanding, there were social anthropologists, cultural anthropologists, linguistic anthropologists, archaeologists, and biological anthropologists. And over time, that's changed somewhat. And now there's kind of a focus on an investigation department, a bioarchaeology and genetics. And I wonder how you continue, like how the foundation has continued to train and recruit and create partnerships with community groups in Guatemala. Like, so is it hiring and working with Mayan community members to have that linguistic capacity? So how have, like, what kinds of long-term relationships have you set up with community groups in Guatemala in order to allow for this kind of careful multidisciplinary work that you do? And what kinds, I think those are, we'll have other speakers in the series who talk about how hard it is to really work with communities in ways that meet their needs and respect their interests. And I think you guys have some real success in that. Can you speak? I know that's not your area, but can you speak to what you think about your multidisciplinary approach over the long term and how that's fostered and maintained those relationships?
Okay, thank you. So first of all, I just want to start by saying that when the families, they demanded to do something for their missings. So the first thing that was created here in Guatemala were civilian organizations or social organizations in charge of collecting the testimonies. They were digging the graves basically with their hands because they were moved by the willing of searching and locating their loved ones. So we have these civilian organizations that have been working in Guatemala since the 1990s, and they have a lot of contact and they have gained the trust of the families, first of all. So we work with these organizations. So they are the bridge between FAFG, which is, let's say, all technical work, and they do like the social part of the work, and that's how we both connect to the families. So basically, the investigation and documentation department, they work alongside with these civilian organizations, and they schedule a visit. They go together to the homes of the families. They collect the interview, they collect the samples, and most of the times these organizations are the ones who also start a legal process, because families sometimes they want justice, not only to recover the remains, but they also want to bring this into justice. So they are the ones who also do that part of the job. And since this is the department who has contact with the families, now or in the recent years, the hiring process also requires people who know Mayan languages. I think especially to Cachiquel and Quiche, which are the main Anishil, which are the main Mayan languages speak here in Guatemala, or the families that we work with. So that's something that we have been creating, having that capability in our staff so that they can relate directly to the family members. Because usually we used to have translators doing that job, but kind of like the relationship or the communication was interrupted. So now we try to do it everything by FAFG staff members. I don't know if that answers completely your question.
It does. Thank you so much.
Okay, so we have a question from Maria. Are you working on or participating in any way in a transnational massive identification project called Proyecto Frontera? If so, could you tell us a little more about the implications, potentials, and limitations of building a massive database with human genetic data between the United States, Guatemala, and Mexico?
Well, I think that the main challenge is to establish who is in charge of that database. Let's say we have a family from Guatemala looking for a family member who wanted to cross the border into the United States. And let's say the body was found in the United States. So here, Guatemala collects the sample of the family, obtains the DNA profile, and it is imported into Guatemala's database. But the US, they have the body, they obtain the DNA profile from that body, and they input that DNA profile into their database. So now we have the DNA profiles, but it is not, they are not on the same database. And these samples were collected or given by the family members to Guatemala agencies. So now is Guatemala agency, does it have the right to share this genetic information with the United States? Or vice versa? Can the United States share that information with Guatemala? So it's about like jurisdiction, administration, legal things, issues that we need to consider in order to share this information. Because as I was saying, everything that the families share with us, any information about the relative of what happened, and even the DNA information has to be treated confidentially. So now if we're sharing that information, are we breaking up that agreement with the family member? And there is another options, because this has happened, like a lot of countries have shared information. They have projects that involve different states, different countries. I don't know if you've heard of ProKids project, for example, that it deals with kids trafficking also. So there are a lot of countries involved. And for example, Emphasis, they have this option where you can share your data in an encrypted file that will only tell you if a DNA match between two databases, if there is a DNA match between two databases, but you won't be able to see any of the genetic information from the outside lab. So that's an option. And you, in that manner, you won't be disrupting any agreement with the donor. So I think that obtaining the DNA profiles, creating a database is not the issue. It's a need. Actually, we need to have these databases, but above that is how we're going to share the information, who is going to upload those DNA profiles, who will administrate that information, how will a DNA match be reported, right? So it goes above like the technical issues. It's more like administrative or issues about the databases.
Thank you. So Diana asks, is there support for families that do not have money to assist in the DNA search?
FAFG doesn't pay any money to family members, not for giving the information or the DNA sample, but it doesn't charge them either. So it is a free process. People from FAFG moves into the communities to collect the sample and the DNA and the information. So families do not need to come from their communities into Guatemala City, into the office to give their information. So we go into the communities, and there is no cost to get their DNA profiles or any part of the process. There was another project called, I forgot the name, but they were in charge. They had budgets from the government, and once a person was identified, they were in charge of giving some remuneration to the family to do the burials and everything else. But it was not part of our organization. It was an independent governmental organization in charge for that.
All right, thank you. So Vivette asks, follow-up question to Lindsay's, perhaps. How has the Guatemala Forensic Anthropology Foundation been able to continue to advocate for the need for a multidisciplinary approach to identification in which genetics is only one of 4 disciplines involved, in face of the growing mystique of DNA? Genetics seems to occupy a lot of the public space surrounding human identification. How can you continue to do this important DNA work and at the same time acknowledge the equal importance of all the other approaches?
That's a very good question, because actually, at a point, everyone thought that DNA analysis was the response to every investigation, not only for human identification, but for criminal cases. Like, everyone was trying to collect DNA samples, do DNA comparisons, and that was the ultimate technique to solve everything. And of course, it has limitations. For example, again, what happens if this human remains, we don't get a DNA profile from it? Then DNA doesn't work. That's it, point, period. It doesn't work. We don't have anything else to do with it. So then the anthropological investigation takes place. Because if they were able to determine the age, the sex, the family reported a fracture in the arm, and now they are seeing it in the bone, the scar left by that fracture, then they are piling up information to do the identification. Of course, this cannot be done in every case, right? This can be done on what we call closed context cases, like the one that one family is looking for one relative and they have a lot of information of what happened or where that relative might be buried at. So their anthropology plays a principal role in that. And there are other identification disciplines, such as using dental records, for example. The thing is that probably because of our context, people didn't have dental records at that time, so we cannot use that. You can also use fingerprints. So there is no most important or better technique. It will depend on the context of your cases and the disciplines that you have available to use, right? And of course, DNA has their advantages, because you're looking for unique information. You can report it with a mathematical or a statistical result. So I think that's why DNA has gained all this fame. But we have to face it that it's not the only technique available and that it has limitations. It does. And for example, the investigation and documentation of victims might not sound scientific department, but if they do not approach the families, then we don't have anything, because we won't have information about the event. We won't have the DNA sample to be compared. We won't know what the family structure is. So even though it is maybe the least scientific department, I think it is the most important department.
Thank you very much, Wendy. I think we are on time. So again, thank you very much for the talk. It was quite interesting and quite complete presentation. So next week we have Belén Sandoval. We are gonna talk, or she's gonna talk, about isotope analysis as this emerging technology and forensics. So, well, thank you very much and see you next week.
If anybody
Thank
wants
you.
to register for more talks, the link to the registration page for all of our talks for the series is below. And Wendy, thank you so much. It's been wonderful to hear about the tremendous work of the Fundación, and thank you for taking the time to be with us today.
No, thank you for having me here. It was a pleasure.
Thank you.
Bye.
Everyone.